Hello everyone. Welcome to the first lecture of Module 8, which is going to be on neuromorphic computing and some of the neurobiological concepts. So the disclaimers kind of remain the same. So let us begin our discussion on neuromorphic computing or brain-inspired computing, and let us try to understand what exactly it is. So what is neuromorphic computing? So the definition says that neuromorphic computing is nothing but a hardware design inspired from biological neural architectures and computations occurring inside the mammalian nervous system or the cerebral cortex. So what this means is we take inspiration from the computation and from the principles of the neuroscience and whatever is happening in your brain or brain of some animals for some particular applications, and then we try to mimic it using the principles of VLSI. So it's a highly interdisciplinary field where principles and knowledge accumulated from neurobiology, computational neuroscience, then computer science, machine learning, VLSI, nanotechnology, etc., are used to design energy efficient systems. So we all know that these biological systems are kind of the most energy efficient systems, and that is why we try to mimic them. So what happens exactly is that in all these modern von Neumann computing systems, like CPU, GPU, or even your DSP processor or your FPGAs, the memory and processor are not identical, or they are two separate units. So every time you have to perform any application, you have to send data via this data bus between processor and memory. Data transfer between the processor and memory not only adds to the latency, but it also adds to your energy kind of dissipation. So that is the reason why in these neuromorphic computing systems or this non-von Neumann computing system, or also cognitive computing, I mean these branches are also known as cognitive computing systems, you have memory and processor as not two separate blocks but as one unit, and that is why they are called non-von Neumann architecture, where processing is done inside the memory itself. And these neuromorphic computing systems form the third generation of computing. So what are the other two generations? So the first generation is kind of the generation which we are utilizing right now. So high-K dielectric based system, nano-S, nanosheet, carbon electronics. So these are the kind of existing architectures that we have already developed. Then the second generation of computing is like 3-5 materials, low voltage circuits and architecture, use of sub-threshold circuits, and then low voltage devices such as tunnel FETs and all those things, or even the spintronics or magnetic devices, spin transistors, magnetic transistors, and so on. So that is what comprises your second generation of computing. This neuromorphic computing device or quantum computing device, these kind of form your third generation of computational devices, and these are like going into the future, we'll be kind of focusing on these devices itself. So now with this kind of introduction to neuromorphic computing, let us look at the historical perspective. So this field is not at all new. I mean, in 1943 itself, McCulloch and Pitts, they came up with a neuron model. And they actually made this model for two weighted inputs and one output, and they were able to implement a simple binary activation function. So I'll get back to this binary activation function in the third lecture, and what exactly are neurons, what exactly are synapses, what exactly are weights, that we'll discuss in this lecture itself. So let us go ahead. So in 1958, Rosenblatt actually came up with the first neuromorphic engine, or perceptron. So this perceptron is kind of one of the basic neural network, artificial neural network, multi-layer perceptron or perceptron, which kind of enables you to classify objects. So I'll discuss that as well in the third lecture. Then this field was kind of stagnant from 1970s, and why so? Because there was the computational capability at that time was kind of limited to process the neural networks for any useful task. So in 1970s, this field remained nearly stagnant. There was no significant development. However, in 1980s, with the advent of these parallel distributed processing systems and backpropagation algorithm, many of these neural networks were neural networks were invented, many algorithms were invented, and they were used for multiple useful tasks. And during the same time, Professor Carver Mead at Caltech, he is known as the father of neuromorphic computing, for your information. He came up with the first silicon retina and neural learning ICs. So in 1980s itself, Carver Mead came up with the silicon retina neural learning ICs, and he actually fabricated these chips in silicon. And even now, his students like Giacomo Indiveri or Tobias Delbruck, Andreas Van Schack, they are the ones who are carrying this flag. They are the ones who are kind of carrying this field on their shoulders. And they're kind of pioneers in this field, even now. Then from 80s to 19, so yeah, Mahawald and Mead's silicon retina, this is something which they developed. So they had this raster network, and the raster network at the joint of it, they had this kind of a silicon circuit, which kind of enabled, vision. So it is kind of a pixel circuit, which they had at these junctions. So then what happened is during 1980s to 1990s, IBM and all the companies are very active in this field. So IBM came up with this neuro-inspired vector classifier engine. So I guess you must be aware of this sector support vector machines, and also IBM in 1990s, close to 1990s, like 85 or 86, it came up with this neuro-inspired vector classifier engine, which is called Zero Instruction Set Computer, or ZISC. So why it is called zero instruction set computer? Because in your von Neumann architecture, you compute with the help of instructions. So you give your computer some instruction that, okay, I want to add two numbers. Then that instruction is fetched from instruction memory. Then it goes to the processor, and then processor processes the summation of those two numbers. And then, the results are again stored back in the memory. So here they do it within the memory itself without the need of these instructions. So that is why it's known as zero instruction set computing. And after this, Intel also came up with this ETAN in like 1989. And this ETAN is nothing but electrically tunable artificial neural network. So it has 10,240 floating gate synapses. And at that time, floating gate synapses were nothing but in the form of EEPROMs. So it had like 10,240 EEPROMs. And Intel came up with this kind of system in 1989 itself. So floating gate synapses, the concept of floating gate synapses is not at all new. In 1989 itself, Intel kind of demonstrated a chip on. At the same time, there were several other demonstrations. So Philips came up with this L-Neuro kind of system. AT&T developed this ANNA or ANNA kind of system. Then Siemens also came up with this SYNAPSE 1 project. And CELT, its Mind 1024 project was really awesome. So they also came up with this, they developed it. And what happened was, in 1990s, there was an advancement in the field of neuroscience as well. So the concepts of this long-term potentiation, long-term depression, and spike timing dependent plasticity, all these concepts were emerging and they were getting a lot of attention, and people started to use these concepts and build neural networks around these 1990s. So even this field, the neuroscience field, saw a lot of development around these 1990s. And in 2000s, this field was transformed by the development or discovery of this memristor. So my advisor was the one, Professor Dmitry Stukov was the one who kind of came up with this memristor, who gave the kind of first demo of this memristor in 2008, while he was a postdoc at HP Labs. And it was from them itself that this field was transformed. It gained a lot of attention. And why so? Because these memristors showed property like incremental step pulse programming, incremental step pulse erase, and which could kind of make them or enable them to be ultimate candidates for synapses. So that was how this field kind of progressed. And this was kind of his paper, The Missing Memristor Found, which kind of came in Nature, and it has been cited like 8,000 times. So it's a very popular paper, as you may also know. There have been several other recent projects as well. So most significant ones are this Blue Brain project by IBM and EPFL, Spinnaker project by Manchester and ARM, the NeuroGrid from Stanford, the Synapse project of DARPA, which is kind of a multi-million-dollar project, the Brain Initiative and the Human Connectome project also, which is happening in US itself, and the Human Brain project, HBP of the European Union, in which, like all the European countries are part of it. And this is kind of a consortium of all the European universities tying their hands and trying to come up with a human brain kind of chip. So these are the most significant or recent projects in this field. So why at all these projects, or why at all this field is so promising? I mean, why at all you want to go for this neuromorphic computing? So the question that I'm asking is, why neuromorphic computing? So the answer is because of the significant advantages that this field of computing provides you. So what are those advantages? So first, it provides you orders of magnitude reduction in the power requirement for performing computations. Second, it provides you an extremely scalable kind of structure. So these memristors can be scaled to dimensions resembling 4f square, where f is your critical dimension. So you can reduce it to the ultimate scaling limit. Then these systems are highly fault tolerant and robust to variabilities. This is very important considering that these memristors or RRAMs or CBRAMs or whatever kind of memory devices we are talking about, they all come with some amount of variations. So unless these computing systems are robust to variations, we cannot even use them. And this property, that it is highly fault tolerant and robust to variations, it makes these emerging non-volatile memories very suitable for this neuromorphic computing. And then another important property of some of these neuromorphic computing primitives is that they are programming free. They don't even need any supervision. I mean, artificial neural networks that need to be trained by supervision. That is, they are called supervised learning algorithms. However, there are some algorithms like spiking neural networks where we don't need to train them. I mean, where we don't need to train them in a supervised manner. So these unsupervised learning capability kind of gives them an autonomous feeling. I mean, they can train on their own just by taking out data from somewhere. So that is another property which makes it very promising. Then it's highly adaptable and reconfigurable. I mean, you can tune your conductance state, your kind of threshold voltage state and all of these emerging non-volatile memory devices at any time during your runtime. So that makes it highly reconfigurable as well. Now this software neural networks and non-von Neumann, although these software implementations and von Neumann implementations are there, so these cannot, really realize the true potential in terms of these parameters. I mean, we already have several software demonstrations, several conventional digital architecture-based or von Neumann architecture-based implementations of these neural networks, but they don't offer these properties which neuromorphic computing does. And why is it so? Why do we, take human brain as reference? So human brain is considered to be the most efficient machine in terms of area, in terms of power. So it only consumes like 20 watts of power, and it only occupies an area of 2 liters, which is very small. Also try to, simulate the human brain. So one of the first attempts in that direction was from IBM. So they kind of attempted to simulate 1% of the human brain. They used this IBM's Blue Gene computer, supercomputer, and the capability required to, simulate 1% of brain is shown over here. So it required at least 64,000 GB of, processing power. I mean, 1 GB RAM is required per processor, and the number of processor that was required was 64,000 to process 1% of human brain, or to simulate 1% capability of human brain. And to process the capability of the cat's brain, they required at least more than 256,000 processors. And the amount of power that it drew from the supply, you will be pleased to know, or I would say you would be shocked to know, that IBM Blue Gene supercomputer draw actually 1 megawatts of power for just simulating 4.5% of human brain. 1 megawatts, which is so huge amount of power. Why is it so? I mean, why is there such a difference? Because your conventional systems like the supercomputer, or even your CPUs, GPUs, they are von Neumann, where your memory and processing are 2 separate blocks. Whereas your human brain is nothing but a non-von Neumann computing system, where memory and computing are happening at the same place at the same time. So let us talk about the difference in the energy efficiency of human brain and your conventional systems. Let us try to quantify the difference in the energy requirement between your human brain and your usual CPU. So in human brain, neurons are kind of the basic building blocks, and the communication between neurons takes place in the form of electrical spikes or chemical spikes. So a neuron kind of takes in 3.84 into 10 power 8 ATPs. So ATP is nothing but adenosine triphosphate, which is a molecule which is kind of decomposed in order to release some energy in the human body. So one neuron requires 3.84 into 10 power 8 ATPs to generate a spike, which then flows through the other neurons, and then it kind of propagates some kind of information, or then you perform some task. So one neuron requires this much amount of ATP, and the amount of energy that is available per mole of ATP is close to 30 to 45 kilojoules. So one spike generation takes 10 power minus 11 joules of energy. Also, if you look at the kind of energy difference between the human brain neuron and your even most advanced kind of systems, processors made from your 5 nanometer technology node, let's say nanosheet FETs, then you'll realize that for generating one spike, the energy that the human brain takes and the energy that this 5 nanometer technology processor takes, there's a gap of at least 1000x. So even your IBM Watson, which is kind of the most powerful supercomputer, that also at 5 nanometer scale, or when it is scaled to even 5 nanometer, that is performing like 1000x times poor in terms of energy efficiency as compared to a human brain. Also, if you talk about the density, the density of neurons is close to, like 1 lakh millimeter square. And there are like 10 per 11 or like, yeah, there are 10 per 11 neurons in your human brain. So the size of one neuron comes out to be as something as 10 micrometers square. So that is pretty huge. I mean, that is not very, that is actually higher than what size of transistors that we have, size of transistors or size of these RRAMs. So we can actually compete with them in terms of area. So if you look at the size, you'll see that till now, whatever silicon neurons we had, their size is actually higher. But with the advent of this RAM and all, we have been able to reach the values of the size of the biological neuron. However, the size of the digital computer is still very high. If you talk in terms of numbers, so the silicon neurons that have been demonstrated so far, I mean, I'm talking about analog neurons or the digital neurons and not the neurons utilizing emerging non-volatile memories. So those silicon neurons have shown energy efficiency of 10 power minus 8 joule per spike. I mean, silicon neurons have shown energy efficiency of 10 power minus 8 joules per spike, whereas their area is close to something like 4000 micrometers square. If you talk about digital computers, so the energy required to generate one spike is close to 10 power minus 3 to 10 power minus 7 joule per spike, which is significantly high. And also, if you talk about the area, so the area of these modern digital processors is close to 263 to 692 millimeters square, or most of these processors are now multi-core, and the area of one of the core is close to 50 to 90 millimeters square. So that is the difference between these biological neurons and the silicon neurons, which are analog or digital implementations or the digital supercomputer. So you can see that there's a huge difference between the two. And it's actually the massive kind of massive parallelism in the human brain. That is, all the neurons are working at the same time. So it's like a huge parallelism, which is making this overall process so energy efficient. And also, if you want to process these like widely parallel kind of systems or neural networks on your digital computers, what happens is there is a high bandwidth which is required to transmit spikes to and fro between the memory and the processor. And this is actually the reason why you have a huge amount of energy dissipation. So what we can conclude is that the true potential of the bio-inspired learning rules can be realized only if we implement them on compatible hardware, or I would say dedicated hardwares which are made or which map those learning rules one-on-one with the algorithm. I mean, so we need to make specialized hardwares which kind of map one-on-one the algorithm on the hardware. So if these algorithms comprise of neurons, synapses, some learning rules, we need to make hardware which can replicate or mimic those neurons, those synapses, and those learning algorithms or learning rules. So what are the main applications where these neuromorphic computing systems are used? So some of the applications are pattern classification, then pattern extraction, recognition. Then they're also used nowadays for machine vision. Then robotics is one of the active areas where neuromorphic computing systems are being used. They're also being used to solve optimization algorithms like the traveling salesman problem, graph coloring problem, max cut algorithm, and so on. They're also used for natural language processing, NLP, which is an emerging area where these neuromorphic computing systems are going into. They're also used for data mining, for big data analytics. They're also being used in data centers. Then I told that some of these systems can be trained even without the help of any kind of supervision, without any supervision. So these form perfect match for intelligent autonomous systems, which you need for space applications or even for war applications and all, such that such as you need autonomous navigation systems, you need autonomous drones, and you also need some prosthetics or brain machine interfacing and all those things. And also nowadays people are already fabricating dedicated neural network accelerators along with their multi-core processing. I don't know how many of you are aware about the recent processor by Apple, M1. So that consists of a dedicated neural network accelerator. So now that we have understood what exactly is neuromorphic, why exactly we want to go for neuromorphic, let us understand some of these neurobiological concepts, because at the end of the day, if we want to make systems which kind of mimic these neurobiological phenomena or neurobiological models, then we have to understand something about these neurobiological concepts. So what exactly is neurobiology? So neurobiology is kind of a very vast and complicated field of science I mean, you can talk about neurons, synapses, spiking potential, action potential, synaptic plasticity, how actual real stimuli which you get from external world is being converted into spikes, action potential, and how this spike information is being processed from the brain. So all these things are covered under this field of neurobiology. So first, let us try to understand one of the basic building blocks of this neurobiological system, which is your neuron. So let us try to understand about neuron. So what exactly is neuron? So neuron is nothing but the basic building block of nervous system. So it's an electrically excitable cell. It processes the information and transmits it with the help of electrical or chemical signals. What are so there are 10 power 11 neurons in your human cerebral cortex, and each of these neurons are kind of connected to, let's say, 10 power 3 or 10 power 4 other neurons with the help of synapses. So what are synapses? Synapses are elements which connect two neurons. So since each neuron is kind of connected to 10 power 3 or 10 power 4 other neurons with the help of synapses, so each neuron consists of like 10 power 3 or 10 power 4 synapses. So this is the kind of neural network organization in your human brain. You have 10 power 11 neurons, and you have like 10 power 14 to 10 power 15 or 10 power 14 to 10 power 15 kind of synapses. And what does the overall intelligence of any organism depend on? So the overall intelligence depends on the number of neurons and how they are connected. So let us see. So you see that this trichoplax doesn't have any neuron and doesn't have any synapse, and it's kind of the most unintelligent organism. As you go down this ladder, you go to roundworm, you go to honeybee, you go to mouse, you go to cat, and finally you reach human. You can see that the number of neurons and number of synapses are increasing, and correspondingly you can see that the sense of hearing, the quality of hearing, smelling, or the quality of intelligence, overall intelligence, that is also improving along the way. I mean, this is not the perfect example of showing you the intelligent organisms, but mind you, he's very intelligent anyways. So the overall intelligence kind of depends upon the number of neurons and their connectivity. Now let us talk about the biological neuron. Let us talk about its structure. It may appear like a biology class to you, but it's actually important before we go ahead and understand how to mimic that neuron into hardware. So what are the different components of a neuron? Let us understand them. So this figure kind of shows you the structure of a neuron, a biological neuron. So it has got three parts. So the first part is your body, cell body, which is called soma. Then there are these dendrites. These are these dendrites, which are kind of branched through this soma. And then there is this axon. So the information kind of propagates through the neurons through this axon. Anyways, I'll discuss about that later as well. So what is this soma? So soma is the cell body, and it contains a nucleus, which also stores your genetic information. Then what is this dendrite? So dendrites are nothing but they are kind of protrusions or extension of this soma, and the information from other neurons that comes to these dendrites. I mean, these dendrites act as receiver of incoming signal from other neurons. They are kind of filaments which are rising out of the soma, and they kind of receive the incoming signals from the other neurons. Now, what exactly is axon? So axon is nothing but a conducting unit, and it kind of carries the electrical signals like spike or chemical spike or action potential. You call spikes as action potential as well. So it kind of carries the action potential to other neuron via synapses. So the other dendrites of the other neuron are kind of connected through these synapses. So you have these synapses which are kind of connecting your other dendrites, the neurons of the other dendrites. And in between you have these vesicles, and then you have this gap which is called cleft. So I'll also discuss how information is being passed from this synapse to this other neuron dendrite and all those things. But you should remember for the time being that this axon is nothing but a kind of conducting channel through which your chemical signal or electrical signal of a neuron passes to the other neurons to which it is connected. So let us now talk about the spike or the action potential, since I talked about it here itself, that it's the action potential which the neuron kind of transfers to the other neurons. So the action potential or the spike looks something like this. So what exactly is the action potential? So it's a rapid, transient, and all or none pulse, like nerve impulse. So either you'll get everything like this, like either you'll get the entire spike like this, or you won't get anything. And the case when you don't get anything is called failed initiation. So I'll talk about that later. So if you look at this kind of spike, what you observe is that its amplitude is close to like 100 millivolts. I mean, it's 110 millivolts or something. and the width is close to 1 millisecond. I mean, the width is not very large. Width is only 1 millisecond. Action potential works is, so we receive a lot of stimulus from several neurons in the form of electrical signals, and there is this potential of the membrane. So let me discuss about what exactly is this membrane. So a neuron is actually surrounded by this plasma membrane, which is kind of made up of this bilayer phospholipids. So this kind of plasma membrane makes it impermeable to flow of other ions. So since it is like impermeable to flow of other ions, there's an inherent kind of concentration gradient between the neuron and its surrounding, right? Because this membrane is not allowing any ions from outside to flow into the neuron, or any ion from the neuron to flow outside of this membrane. So because of that, there is an inherent concentration gradient, and because of this, what happens is your membrane in equilibrium or in an unperturbed state, that membrane stays polarized at a resting potential of minus 70 millivolts. Now, since it's at a resting potential of minus 70 millivolts, you can also receive neuron spikes from several other neurons, and they are kind of integrated at the neuron. So this I'll discuss later, how this action spike potential is kind of generated. So the thing that you should remember here is, so there is this firing threshold, which is close to minus 55 millivolts, and there is this spike or stimulus coming out of all these incoming neurons to this neuron under consideration. And if this stimulus increases in magnitude than minus 55, there would be this action spike potential that will be generated. However, if this overall stimulus is less than minus 55 millivolts, or if overall stimulus is not able to change the membrane potential by increase the membrane potential above minus 55 millivolts, there won't be any firing. And this kind of situation is called failed initiation. So we'll discuss about it later in the next slide. Also, in the membrane, you have something called ion pumps or voltage gated ion channels. So these are special kind of protein membranes, and these kind of allow the flow of ions in and out of this neuron. So what are those ions which can flow in and out of these neurons? So those are sodium, potassium, calcium, and chlorine. And these kind of flow of these kind of ions helps in propagation of this action potential from one neuron to another via the synapse. How so? We'll discuss in the next slide. And these ions are very much responsible for the propagation of action potential or information throughout your brain. And that is why when you have deficiency of these ions, what happens essentially is that your brain stops to work. I mean, you can't work very well. I mean, you start hallucinating or something like that. Now let us discuss how this kind of action potential transmits between the neurons through the synapse. So what exactly is a synapse? So a synapse is nothing but a region where your action of a neuron gets closer to the dendrite of the other neuron. So it's like the action potential flows through the action of one neuron, and then it flows to the dendrite of the other neuron. And synapse is a region where one neuron interacts with the dendrite of the other neuron. So it acts like a communication channel between the neurons, or a conduction medium between the neurons. So let us talk about some terminologies. So a neuron which is sending the information or spike train or action potential, that is called a presynaptic neuron, while a neuron which is kind of receiving the information or the spike train, that is called postsynaptic neuron. Now this information or this action potential or this transmission of action potential can be electrical, which is very fast, or it can be chemical, which is actually slow. So electrical kind of transmission or electrical signals, they happen like their transmission delay is close to 0.2 milliseconds, while for the chemical or the chemical signals, the transmission delay is close to 2 milliseconds. So your defensive reflexes, where you need to act very fast, they are mostly dependent upon this electrical kind of mechanism, electrical transmission mechanism. Whereas your chemical mechanism, these are kind of it's like they're more responsible for the sense of thoughts, or they are mostly present within your central nervous system, and they're mostly responsible for thoughts or perception or anything like that. Now let us talk about how this action potential travels through axon, and then how this information or this action potential is transferred to the other neuron. What exactly happens when the information transfers to the other neuron? So this action potential actually traverses through the axon and to the postsynaptic neuron. And once this action potential reaches that postsynaptic neuron, what happens is there is a depolarization, electric depolarization of the membrane at synapse. So what this leads to? This opens up the ion channels which are permeable to calcium ions. And now so what happens is this action potential is coming through axon, and it is kind of reaching this point. And then what happens is, because of this action potential reaching this point, there is a depolarization of this membrane. And because of this depolarization, what happens is there is this ion channels which are permeable to calcium, they are opened. So some ion channels are opened, and as such calcium ions are able to enter this presynaptic vesicle. So these are your vesicles. So your calcium ion is able to enter these vesicles, and once these calcium ions are able to enter this presynaptic neuron, what happens is they activate this protein in the vesicles which release this neurotransmitter. So it's these neurotransmitters which are having information. So what happens is, the moment your action potential is reaching here via synapse, they kind of depolarize this membrane. The calcium ions are now allowed to flow into this presynaptic neuron, and once they flow into this presynaptic neuron, they activate these proteins near the vesicles. And once those proteins are activated, these neurotransmitters inside those vesicles, they can now flow out. And then these neurotransmitters through these vesicles, which are now flowing out of this presynaptic neuron, what they do is they travel through the region between the neurons. This region is called a cleft. So this is one neuron, and this is your dendrite of the postsynaptic neuron. This is your axon of the presynaptic neuron, and this is your dendrite of the postsynaptic neuron. So these neurotransmitters have been now released by these presynaptic neurons, and these travel through this cleft, that is the gap between these two neurons, and they reach where? So they reach the dendrite of this postsynaptic neuron. Now in the postsynaptic neuron, these kind of neurotransmitters, the transport of these neurotransmitters is diffusion basically. So these kind of diffuse through this cleft, and then they reach this postsynaptic neuron, the dendrite of the postsynaptic neuron. Now dendrite of this postsynaptic neuron has several receptor sites consisting of several receptor molecules. Now these neurotransmitters simply bind to these receptor sites and activate them. So what happens is, so these binds to the receptors, these neurotransmitters binds to the receptors in the postsynaptic membrane and activate these receptor sites. Now activation of these receptor sites, that opens up ion channel. Okay, so these ion channels are now opened up in the postsynaptic cell, and they allow ions to exit or enter into this postsynaptic neuron membrane. Or the opening of this ion channels, what this leads to is, they leads to exchange of ions between the postsynaptic neuron and its surrounding. What this leads to is, this leads to a change in the membrane potential of the postsynaptic neuron. So the postsynaptic neuron is not in its resting position anymore. So the membrane potential has changed. Why it has changed? Just because these neurotransmitters have activated these receptor sites, and the moment these activate these receptor sites, these like this postsynaptic neuron can exchange ions, and because of that, its membrane will either depolarize or hyperpolarize, leading to a change in its membrane potential. So this leads to what? This leads to a postsynaptic potential change. So this kind of membrane potential is called, so the membrane potential of this postsynaptic neuron is called postsynaptic potential, PSP in short. So this kind of action, this kind of binding of the neurotransmitter leads to a change in the PSP. So if this postsynaptic potential or the PSP depolarizes the membrane, or it increases its kind of conductivity, it's called excitatory PSP. And if this kind of postsynaptic potential change hyperpolarizes the membrane or reduces its conductivity, it is called inhibitory postsynaptic potential. So on the basis of whether the synapse leads to an excitation or leads to an excitatory PSP or inhibitory PSP, it is kind of classified as excitatory excitatory synapse or an inhibitory synapse. Now, what does these neurons do? I mean, this is with respect to time. So EPSP simply leads to increase in the conductance. IPSP simply leads to a reduction in the conductance. Now, what does the neuron do actually? So neuron does nothing but integrates all the incoming postsynaptic pulses from all the dendrites which are connected to the other neurons. So all the presynaptic neurons send some amount of action potential to the postsynaptic neuron. It kind of integrates all these PSPs, like some of these PSPs would be excitatory, some of these would be inhibitory. But what this neuron does is it integrates all these EPSPs and IPSPs. And the action of all these IPSPs or EPSPs changes the membrane potential of the postsynaptic neuron, right? So effectively, if the summation of EPSP minus the IPSP is greater than minus 55 millivolts, there is a spike generation in the postsynaptic neuron, as I told over here. So if you look at this slide, so as I told that all EPSPs and IPSPs are kind of integrated over here. If the change in the membrane potential because of integration of all these kind of actions taking place is larger than minus 55 millivolts, the postsynaptic neuron will fire this spike. Otherwise, if it is less than this minus 55 millivolts, it won't fire. So let me just go ahead and tell you that. So if this is greater than minus 55 millivolts, there's a spike generation in the postsynaptic neuron. That is, if it depolarizes the membrane beyond this minus 55 millivolts, there is an action potential or generation of a spike in the postsynaptic neuron. And it must be kept in mind that only if this EPSP minus IPSP summation of this, I mean, integration of all of this is greater than minus 55 millivolts, there would be firing of a spike. If the membrane potential due to summation of all the or accumulation of all the stimuli is less than minus 55 millivolts, there is no spike, and this is called failed initiation that I already told. So in case this membrane potential is remaining below minus 55 millivolts, that is, if your IPSP is kind of dominating over EPSP, then there is no spike, and it is called a failed initiation condition. The moment this failed initiation condition is achieved, what happens is the ion pump and the voltage gated ion channels in your membrane, they kind of restore the membrane potential to the resting potential. So you saw that, you know, the potential increased somewhat due to stimuli, but it again came back to its resting potential in event of your failed initiation. So that is what happens. And the best part about this neurons is, or the interesting part is, that the release of this neurotransmitter onto the dendrite of the other neuron is actually probabilistic. And what this leads to it leads to a stochastic kind of spiking. So even if your EPSP minus IPSP is greater than minus 55 millivolts, you cannot be sure that, you know, there would be a spike, because the release of neurotransmitter is probabilistic. And also, this is one kind of neuron. I mean, this is one kind of neuron which is very commonly present in your hippocampus, but there are other neurons as well present in your brain or the brains of other mammals as well, where they act as, you know, a pulse frequency processing element. So you give it a spike train, the neuron will also convert it into a spike train of, let's say, double frequency, half frequency, or something like that. So it will act like a pulse frequency processing element. So that is also a role of some of the neurons in mammalian brain. So it's not like all the neurons are performing the same application. This is the application which is performed by most of the neurons, but there are also some neurons which kind of act like a pulse frequency processing element. There exists, you know, several kind of these behavioral models which represent this kind of integration capability of your neuron. And these electrical models, they're called as neuron models. They do not capture the details at the intricate details of, you know, individual ion channels, but they're mostly behavioral models. And the complexity and details which are included in these behavioral model depends on the application for which you are going to use them. For most neural networks, be it spiking neural network or artificial neural network, a very simple kind of a model which just represents this neurons as, you know, a kind of integrating thing like a capacitor, that works really fine. So let us go ahead and look at few of those simple behavioral models. So one of the simplest neuron model is the integrate fire model, which was kind of discovered in 1907. So it kind of takes into account of, like, it represents these neurons as capacitive elements, and this is driven by, you know, the differential equation for capacitance, which is IT is Cm PVM by dt, where the Cm is nothing but the capacitance of this neuron, and the neuron membrane actually here acts like a capacitor, and it integrates all the currents and fires a pulse or spike only if your Vm is greater than Vth, or the membrane potential is greater than Vth, and this Vth is nothing but minus 55 millivolts for real biological neurons. Another, like I would say, one level up this model, where they also take into account the leakage due to the drift of your ions in your membrane, considering that membrane is not a perfect insulator, but there are some ways in which ions can move out of it. That is called a leaky integrate and fire model. So there what they do is they just include this leaky term, which is minus Vm by Rm. Rm representing the path, the resistance of the paths from which the ions can leak. This just takes into account that your membrane is not perfect insulator, and it is just one level up, or one of the advanced model as compared to your simplest model of neuron, which is integrate fire model. And this is most common used model for spiking neural nets and all. Then there are some advanced model as well. For instance, this Hodgkin-Huxley neuron model. Here, instead of solving only one differential equation, you need to solve 4 different differential equations. So the circuit, effective equivalent circuit, looks something like this. So here also we have Cm, which represents the membrane capacitance. Then you have these non-linear conductances Gn and Gl. Here Gn and Gl, which kind of represents your different ion channels. And these are non-linear conductances, which are function of voltage as well as time, as you can see over here. Then, as I already told, that there is an electrochemical potential gradient between the membrane and the surrounding, and that electrochemical potential gradient is kind of modeled as batteries. So En and El kind of model that electrochemical potential gradient. And then we have the ion pumps. As I told you, that these ion pumps help bring it back to the resting potential the moment there is a failed initiation. So that is modeled as a current source, which quickly discharges this Cm and brings it back to its resting potential. So this is also a kind of model which is very advanced. I mean, this Hodgkin-Huxley model is an advanced neuron model, but for all practical applications like SNN or ANNs, for SNNs typically this LIF model works perfectly.